Aloha. Thank you all so much for coming out this evening. What I wish to do tonight is basically two things. One, introduce to you the idea of the importance of parrotfishes or uhu and sea urchins, and also basically all fishes that eat seaweeds, to conserving our coral reefs in the face of climate change. And then secondly, introduce to you a long-term study that my lab has started on encouraging the herbivores to live on degraded reefs and help them recover after coral bleaching, which is all part of this story. My co-authors are my graduate students, all PhD students at UH Manoa. Coral reefs are such a blessing to Hawaii. They are valuable to us in a huge number of ways, but just quickly reviewing those, they protect our coasts from coastal erosion, which is becoming more and more important as sea level is rising. They also produce great surf spots. Of course, seafood. This is a classic painting from the Honolulu Museum of Art, showing the bounty of the sea that comes from Hawaiian reefs. Of course, recreation and tourism, Hanauma Bay being a prime example of that. And what most people don't realize is that we're increasingly getting extremely powerful medicines from coral reef organisms. This is just a few examples here from very humble organisms. Here from a simple sponge is a new drug that is being used to treat breast cancer. Here from a sea squirt for treating leukemia. And here from another sea squirt that treats ovarian cancer. Very complex molecules, as you can see there in the structures, produced by these different organisms for various reasons, but the pharmacologists are finding increasingly that these compounds, unique compounds produced by coral reef organisms, have medicinal properties. And unfortunately, we're losing reefs faster than we can screen many of these species. So coral reefs are just the bounty from the sea for us. But unfortunately, our reefs are starting to become increasingly stressed. This is Molokini in 2015 during the last mass bleaching event. Coral bleaching occurs when the water gets so warm that the mutualism, or the mutually beneficial relationship between the coral and little single-celled plants that live inside the coral, breaks down. So the coral expels those single-celled plants, losing its color, which is why it's called bleaching, not really bleach per se. But once that's broken down, that relationship, the coral cannot survive for long. But if the water stays warm, those bleached corals die. And with that, the reef dies. So how can we stop this? The ocean is getting warmer and warmer and warmer. Let's look at the patterns here in Hawaii. The first coral bleaching ever noted in Hawaii was in 1996, just at the beginning of one of the warmest years on record then. Then in 2002, scientists noted the first coral bleaching in the Northwest Hawaiian Islands, which eventually became Papahanaumokuakea. In 2014, the first mass bleaching occurred. This was primarily around Oahu, especially in Hanauma Bay, but fortunately the corals recovered quite well, and that was not a major mortality event. But then just the following year, 2015, was a second major bleaching event, which hit mostly the Big Island and Maui Nui, and there the corals have died. It's a really bad situation right now for particular species of coral in these areas. And so this is just going to be getting worse and worse. The data just came out today, announced by the federal government, that 2017, 2016, and 2015 are the 3 warmest years on record. And we're just entering a phase of increasing warming and increasing bleaching. So projecting forward, scientists have recently predicted that by 2040, we'll have coral bleaching every single year. And worldwide right now, the bleaching is recurring on average every 6 years. And that's scary to the scientists because typically after corals die, it takes at least a decade for full recovery. So if you're on average, you're getting bleaching every 6 years or every year, the reefs can't recover. So what can we do? That's one thing the scientific community is facing right now. What can we actually do besides just documenting the decline? Well, there's a number of things we can do, but they're all under a heading called resilience. That is, enhancing the natural ability of our coral reefs to resist bleaching, or when they do bleach, to recover from bleaching and come back for future generations. So what we must do before the reefs completely die and look like this is Kahekili in Maui before the herbivore recovery area was put in place. This was killed by injection of nutrients from injection wells that leached onto the reefs and caused the algae to displace the corals. To fight this, ultimately, we need to stop climate change from happening. We need to reduce carbon emissions. Most of the world has gotten on board with doing that, and hopefully there'll be changes forthcoming, even here in the United States. But locally, there's a whole bunch of things we can do. A very important factor is water quality, from keeping sewage and pollutants from getting onto the reefs, the sewage especially, and fertilizers and sediment from runoff. First, help algae grow, but also the sediment smothers corals. So if we can keep our watersheds clean and keep the water that's running off onto our reefs relatively clean, that's going to be a huge, huge benefit right there. But what I'm going to focus on tonight in the research of my lab has to do with preserving the seaweed eaters and why they're important. This is a large uhu or parrotfish. So let's look at the role of the seaweed eaters on the reef. What happens is seaweed eaters help corals indirectly, and they do that by controlling the main competitors of corals. So if you kind of follow this loop here, sea urchins, parrotfish, surgeonfish, and chubs have very strong negative effects on seaweeds. They keep the seaweeds cropped low. And that's important because seaweeds, if they're given the chance, can outgrow corals. They grow faster than corals. They carry microbes that can attack the corals. They sequester sediments that can smother the corals. So by controlling the seaweeds, there's reduced competition with the corals for living space on the reefs. If the corals benefit, they in turn provide shelter for the sea urchins and parrotfish, and you get a nice positive feedback loop going on a healthy reef. Corals are growing, they provide shelter for these grazers, the grazers thrive, they keep the seaweeds under control, and everything's happy. But picture now this system if something happens to the corals. If the corals die and break down, like I showed in that last slide, then there's no shelter for those fish to live in and those urchins. So the populations of those grazers declines, and then the seaweeds have a heyday. The corals are dead, they're not competing with the corals anymore, there's no grazers grazing them, and you end up with what the scientists call a phase shift from what used to be a coral reef to this dead coral rubble covered by seaweeds, which is of benefit to virtually no one. So how can we enhance these grazers on reefs? And that's the study that my lab is now doing. We're actually using an ancient tool that the Hawaiians used many times called imu. And imu are basically, as you can see in the lower right corner there, just piles of boulders. Piles of boulders stacked up that create shelter for fish, shelter for urchins, concentrating those animals in an area. And if those animals are grazers, then they'll keep the seaweeds grazed around that particular area. The Hawaiians used these to aggregate fish in areas where there weren't reefs, out on sand flats. But we're using this tool in a modern version to see if we can enhance sea urchins and parrotfishes locally. So here's the modern version of an emu. All it is a stack of concrete blocks held together by stainless steel banding. Now you'll notice I have two different styles of these things. One style has all those holes open, holes of three different sizes, everything from little tiny sort of square half-inch holes for little tiny baby fish and sea urchins to live in, and then a slightly larger size for the juvenile fish and urchins, and then big holes for the big fish and urchins. Places to live and seek shelter. That's what these things like. The reason we also have the modules that are where the holes are covered is because we're doing this in an experimental context. Yes? It's about a cubic meter, a cubic yard. Those are basically just standard 8 by 8 by 16 concrete blocks. So what's the experiment we're doing? What we want to do is test predictions of the hypothesis, if you will, that providing shelter for seaweed eaters indirectly helps the corals, as I showed you before. And if you can imagine a reef that's died and collapsed, there's no shelter there. So if we put these things out, will they work? So here's the idea, as you can see in this diagram. With these, I'm calling these modules two different kinds. There's open modules, which means the holes are open, and closed modules, where the holes are closed. And the predictions are that the fish and urchins will colonize these modules that have lots of holes and not so much on the ones without holes. Then in turn, with all that grazing intensity, there'll be relatively few seaweeds where there's lots of grazers, relatively lots of seaweeds where there aren't, and ultimately the corals will thrive where there's very little seaweed and will not thrive so much where there's lots of algae. So those are the original predictions of our experiment to test whether it really is shelter that's running the show and indirectly facilitating the corals by bringing in the grazers. Okay, everybody with me? Okay. So another important component here is how intense fishing is taking place, because if people are removing the fish, it doesn't matter whether you provide shelter for the fish. So we actually wanted to compare a relatively degraded reef where there's been lots of fishing, and this is Waikiki, compared to a relatively healthy reef, which is what brought us to Hanauma Bay. And so the overall experimental design that we were able to get approved by 14 city, county, state, and federal agencies over a 2.5-year period is this. 3 modules of each type in Waikiki and 3 modules of each type in Hanauma Bay. Now, we didn't put these on the reefs because we didn't want to damage the corals that were already there. So these are out on the sand flats. I don't know if I can, there's no big map right out here, but if you go out sort of toward the mouth of Hanauma Bay, there's a very large sand flat at about 50-foot depth. That's where these modules are located. So snorkelers never see them, and most divers don't go out over the sand flats. So they're not bothering anybody. And similarly, there's a large sand flat off the Halekulani Hotel off Waikiki that we're also using. And there's a 100-foot distance between these things. We have 3 of each kind because for statistics, you need what's called replication. And this was the maximum replication I could get. So we're calling this the Coral Resilience Module Experiment, or the CREM experiment, if you will. That is, will providing shelter for the grazers enhance the corals? So let's look at the results. This experiment's been running not quite a year and a half so far. We set up the experiment in the summer of 2016. And these are just typical shots of these modules at 8 months in the top and 14 months at the bottom. And what you can immediately see is huge differences in the amount of seaweeds. And that's because there's almost no grazers on the modules in Waikiki. There are neither fish nor sea urchins colonizing these things because they're just not available and they're just not settling anymore. Whereas in Hanauma Bay, sea urchins love these things. And these aren't sea urchins that crawl out from the reef. These are little tiny baby sea urchins that settle from the plankton as larvae and really like these modules and keep the algae grazed bare. So it's not exactly what we predicted, because it didn't really matter whether there are many holes or not. There's more urchins where there's lots of holes, but there's still urchins on the modules that don't have holes. And in Waikiki, it doesn't matter because there's no grazing, there's no grazers there at all. And the corals are starting to settle. And this is what we're just loving to watch and follow through time. So these are the two main species, parrotfishes, which is a sort of a finger coral, and Pocillopora, which is more cauliflower coral and things of that sort. And you can see these are really babies. The 2 centimeters there in the lower right is basically an inch. So these are the corals not long after they settle and just starting to grow. And the percentages you see are the overall grand percentages that we've seen after almost a year and a half. So most of the corals are Pocillopora, and about 13% are parrotfishes. And what's most fascinating to us is we're actually seeing more corals growing on the modules with seaweeds on them in Waikiki, 76% of all the colonies we've counted, compared to Hanauma Bay. And this is fascinating us. And what we believe might be happening, but it's too early to tell for sure. I'll show you some graphs relative to this, but it may be that there's so many urchins on the modules in Hanauma Bay that they're actually removing the baby corals as they graze. You can see just little flecks of brown around those lower corals, which is where the grazing took place, right up to the edge of them. Whereas in Hanauma Bay, whereas in Waikiki, they settled more broadly, but they're increasingly getting surrounded by seaweeds. And so right now our prediction is that ultimately Hanauma Bay is going to be better off, because the corals that do survive are going to grow just fine, whereas those in Waikiki are going to end up being smothered as the seaweeds outcompete the corals. But it's a little early to tell.
What is Pocillopora?
It's like cauliflower coral. It's the small branching corals that we see on our reefs. And no, it's not. It's not cauliflower coral. I'm sorry. Parrotfishes is cauliflower coral. Pocillopora is, I'm trying to remember the common name of it. But it's a, yes, thank you. Yes, parrotfishes is the finger coral.
What is it?
Lace coral? Yeah. I wish I had, next time I give this talk, I'm going to bring some chunks of coral. That'll help, because these are just the little babies. Now, one thing that has happened in Hanauma Bay is we have rare coral species, relatively rare, that don't occur in Waikiki. Waikiki, we've only gotten 2 species so far. In Hanauma Bay, we've also gotten a coral called Cyphastrea and Montipora, which is also a fairly common coral elsewhere in Hawaii. So Hanauma Bay has the most species of coral so far. What we like about this experiment is everything went out in the ocean at the same time. Same materials, same depth, same conditions. So the differences are due either to the structure of the modules or to the location. And what we're seeing is location is very, very important.
How far out?
How far out? They're out toward the mouth of the bay in about 50 feet of water. In the sand, not on the reef. So what we're doing is following the fate of individual coral colonies through time. And this is just 2 examples of parrotfishes. The 1 centimeter scale bar there is about ½ an inch for scale. So you can see going across the top, that's exactly the same colony as it's starting to grow on the module. And down below, with the Pocillopora, you can see that it's starting to grow branches that are growing out. And we're following hundreds of these individuals. So we will be able to know through time which get smothered by algae, which get eaten by something. In one of the modules at Waikiki, we had a crown of thorns starfish appear and eat most of the corals on there. but we can really follow the fates and see exactly what's happening with these corals. So let's look at a few graphs. I hate to show graphs, but they kind of show a pattern here. So on this axis is time, okay? So we started in August 2016, August 2017, all the way up to this last September. And the y-axis is the number of fish that we see on the modules. And this is looking at all fish that are grazers and fish that aren't. And what you can see very obviously is that only the modules that have lots of holes in them in Waikiki have attracted fish. The modules in Hanauma Bay are not attracted to fish. And we believe the reason is that these modules are surrounded by beautiful, healthy coral reef. Why go live out on concrete blocks when you can live on beautiful corals? Whereas off Waikiki, there's almost no reef. I showed you the picture. It's just all collapsed, dead bench. So if there is shelter available, that is the open modules, the fish just swarm to them. And that's what we see here. The two species we show up there are two of the most common species. To Ape, they introduced snapper and a soldier fish. Now, if we look at just the fish that eat seaweeds, we see very few. It's the same kind of graph, but look at the numbers here. Right in the middle of this graph is 2 per module. Almost no herbivores are settling on the modules. But what we have noticed is that the fewest of the grazers are settling on the closed modules at Waikiki. That is, they're just not attractive to them. And you can see they kind of come and go on the other modules. Those lines are crossing each other all the time. So we're not getting much in the way of grazing fishes so far into the experiment. This experiment, by the way, is slated to last 10 years because we want the corals to grow all the way up. The main herbivores we see, or the main grazers, are surgeonfish, and there's 2 of the typical species there. We've seen almost no parrotfish, no uhu. And that's really quite concerning to us because uhu are the best at removing seaweeds. And they have been so severely overfished on this island that even if you provide shelter for them, not many show up. Yes?
What is ANOVA?
I'm sorry? Oh, no, that's the statistical analysis we used.
Oh,
Okay, it tells us whether these patterns we see are probably true or not, based on how variable they are. So the little lines you see with each point is how much variance there is in the modules, how different things are, natural variability. Really didn't want to get into that, but thanks for asking. Yes. Well, you can see here, these are our survey dates, okay? So we didn't go out that often at first because not much was happening, but then we're typically going every month now. Yes. Okay, so the fish aren't doing much is the bottom line. But sea urchins are really running the show, as I told you earlier. So this is the same kind of graph through time, looking at the number of sea urchins, about 6 different species or so on these modules. And you can see that the open modules, the modules with holes in Hanauma Bay, are just covered in urchins. The urchins love these things. Up to 50 urchins per module. It's a huge amount of grazing. You can also see that they peaked during the summer and are starting to decline. And what that means, and what we've seen, is that during the summer months, when there wasn't much in the way of south swells this year, lots of baby urchins settled and started growing. But as we start going into the fall and getting windy conditions, they were either leaving or getting knocked off and rolled around. Again, time will tell. We're going to look at long-term patterns here. And now the corals themselves, very, very clear patterns. So we saw no corals on these things for the first 6-7 months. I mean, they just hadn't settled, and either that or they were so microscopic we couldn't see them. But you can see that regardless of which type of module there is, the corals are gradually accumulating through time. way more corals in Waikiki than at Hanauma Bay, but nonetheless, the corals are accumulating regardless of the treatment. So Waikiki has far more corals, and there's really no difference between closed and open, although the averages are different. And Hanauma Bay has fewer, but they're still growing there. So this is where we were concerned about, is this being caused by the fact that there's relatively more urchins in Hanauma Bay? Are they removing the small corals overgrazing? Time will tell. This coming year, we're going to actually start doing some experiments with urchins off Waikiki and manipulating their abundance to see if they really are running the show. Now, one pattern that struck us that I want to show you, and I promise it'll be the last graph, is at the very peak of urchin abundance, where there were, upwards of 50 urchins per modules that had holes. We examined the relationship between the number of sea urchins there on the x-axis and the number of baby corals that were settling. And so each of these dots represents one of the modules at those times. But there's 12 of these points overall, and you can see very striking differences between these two sites. At Waikiki, there were basically no urchins that ever settled. We never saw more than one or two, but a huge range of baby coral recruits coming in, ranging far higher than we ever saw at Hanauma Bay. Whereas in Hanauma Bay, the number of corals increased with the number of urchins initially, which is what one would expect, right? Less seaweed, more space for corals. But then at the very highest abundances of urchins, the number of corals drops down. So it's almost like too much of a good thing. If there's too many grazers, they actually inhibit the corals, but they're good for the corals at lower densities. Yes?
In the previous slide.
Previous slide. Oops. There we are.
There you go. There's a delay of a year before you see much. Does the surface have to accumulate anything else before corals will start growing on it?
Yes, good question. So the question was, why did it take 8 months before any corals showed up? There's a couple reasons for that. One is the larvae have to be there. So depending upon when the corals spawn and when their larvae are available.
That didn't look like spawning. They normally summer spawn.
Exactly, and that's where we come to the second reason. The second reason is that these surfaces that we put in the water were fresh, clean concrete. Now we soaked the concrete in fresh water and got all the chemicals leached out of them before we put them in the ocean. But they were just virgin substrate. It was like bare rock. And what happens is you have to get a microbial film forming and chemical cues that attract the corals. So that's just a delay in the right conditions for the corals to settle that we're seeing there.
Thank you.
Okay, so getting back to the predictions, it's really too early to tell whether this ideal pattern that we laid out before will ultimately take place. And we didn't expect things to happen that fast. It's going to take time. But so far, if we look at all these different predictions, we saw that in terms of the prediction that there'd be more grazers on open modules compared to closed modules, definitely true for urchins at Hanauma Bay, but basically no fish have settled on the modules in Hanauma Bay. And again, we're assuming that what's happening here is the fish are just choosing to live on the beautiful natural reefs that live in the area. On the other hand, in Waikiki, where there basically is no natural reef left, there lots of fishes are colonizing, but the only grazers are a few species of surgeonfish, no uhu yet. And what about the urchins? Why aren't the urchins colonizing in Waikiki? Well, there's a couple reasons, but we think probably is what's happening is we've noticed big triggerfish that are hanging out on these particular modules, and triggerfish will eat sea urchins. And in fact, there's been times when we found a sea urchin on a module, come back the next time, and we see spines. Something happened to that urchin. It looked like it had been eaten. We're not seeing those big triggerfish in Hanauma Bay. Again, time will tell. These are just early patterns so far. What about the seaweeds? Well, it's not really following this pattern, because regardless of whether there's many holes or no holes, we see almost no seaweed in Hanauma Bay. That's because sea urchins don't seem to care whether there's holes or not. Again, the fish aren't really part of the system. In Waikiki, we see seaweeds everywhere because there are no grazers anywhere to speak of. So that, again, not really following the pattern. Mother Nature always throws curveballs at us. And then finally, there's less coral settling at Hanauma Bay, but it appears to be in a healthier state because there's no seaweed growing around it. There's more coral that's settled in Waikiki, perhaps because less urchins removing the babies, but they're at risk because seaweeds are growing right up against them and starting to grow over them. But again, this is only 14 months worth of data. We're just getting started. Corals grow very slowly, maybe a half an inch a year. So stay tuned, and hopefully I'll be back another time to show more definitive results. And I just want to finish with this slide. The Reverend Sam Kapu was generous enough to come out and bless the experiment at the very beginning. He's here with my graduate students and me, my wife Sherry, who's here today, and Tara Hirohara, who's the director here at Hanauma Bay. And it was one of those incredibly beautiful days with bad weather on either side of it. We scheduled it that day. It was just gorgeous. So it was really a wonderful event. And if you don't get all your questions answered or you want to check in with me in the future, this is my email address. Happy to answer any questions. Thank you. Yeah.
Such a small number of stations, knowing how smart predatory fish are. You taken some time-lapse photography to see if some fish have decided that this is going to be part of their game to visit the certain ones.
Okay, so the question is, given that predatory fish are smart and can learn what these things are, have we basically sent up a smorgasbord for predators? And we've done these experiments before in the Caribbean region, and what we've seen is that on the modules that have lots of holes, the prey fish are relatively safe. That is, the predators don't tend to go in the holes after the prey. But the exception is moray eels. And one thing we have noticed is moray eels colonizing the modules in Waikiki. Not in Hanauma Bay, but remember that's opposite the pattern. Hanauma Bay, there's just not many fish, period. In Waikiki, despite the fact that there are moray eels colonizing the modules that have holes, there's also lots of other fish. So far that pattern you pronounced doesn't seem to be happening.
God is playing tricks on you?
Oh, Mother Nature plays tricks on biologists all the time. Believe me, we set up this beautiful experiment with these very clean predictions, and everything's just strange. So we very much look forward to seeing how things develop through time. Well, it brings humility is what it does, to tell you the truth. Yes.
You know the Hawaiian name for urchin? I've been trying to think of it.
We had this discussion because I found one word which I didn't memorize. I'm sorry. I found one word for urchin, and Mehealani said, "Well, I think there's multiple names for different species of urchin." So
the urchin he showed up there on the screen is bana.
What?
Bana.
Mana.
And then the other, the pencil slate urchin has a, yes. And then the collector urchin, or sorry, not the collector urchin, but the rock boring has a different Hawaiian name, but also I'm trying to think of it, the helmet shield
That's what
urchin.
I know.
Yeah.
The Vana, I'm very familiar with Vana, and Vana did not have all those thingamajigs. But anyhow, when I was growing up on Kauai,
Yeah.
8 years old, we used to catch the urchins and we put it in bags and just shake everything off. And then we would eat it. And it was delicious.
Oh yeah, there's a very pronounced urchin fishery in many parts of the world, with the roe, the eggs of the urchin, called uni in Japan, are very, very popular. Yeah. Yes.
Dr. Hixon, relative to temperature change, why do the plant organisms at a certain temperature leave the coral?
So why does coral bleaching take place? Basically, that's being studied very deeply right now. And my understanding of it as a fish person is that relationship is very temperature dependent between the single-celled plant-like things that live in the coral and the coral. Those two species feed each other. Okay, the coral feeds its nitrogenous wastes to the algae and fertilizes the algae. The algae in turn photosynthesizes and gives some of its carbohydrates to the coral. So it's mutual feeding that takes place. One of the hypotheses is that as the temperature increases, the algae switches from giving to taking. It sort of becomes a parasite to the coral. And at that point, it's not worthwhile for the coral to keep the algae, so it expels the algae.
Now, if the algae is more active, perhaps, I'm just guessing.
Yeah.
That if the temperature goes up, I'm thinking, I don't know why I'm thinking the sunlight goes up, but if the temperature goes up, they become more active and then they start taking. Is something like that?
It's something like that, yeah. But the problem is the mechanisms are at the biochemical level. And, you know, a coral is an amazing thing. It's not one species. It's those single-celled plants. It's the coral animal itself, and then it's a whole bunch of microbes all living together. And so it's really a community. What we see as a coral is really a community of organisms, most of which are single-celled and microscopic. And so teasing apart all those interactions is a place of very active research right now, and we don't have all the answers yet.
Is it true that if they stop taking, don't have any more nutrients, is that when they leave?
They're actually expelled by the coral.
They're expelled.
They're expelled. So it's really a desperation move by the coral to survive. It's like, this thing that used to be feeding me is out now actually taking from me and is inhibiting me. In any case, the relationship breaks down. And so, you know, get rid of it. It's not something I want, even though I will starve without it. And so what happens is, if the temperature returns to normal fairly quickly, there's usually enough cells left in the coral that they repopulate and the coral recovers. And this is what happened in 2014 in Kaneohe Bay. We saw mass bleaching, but almost all those corals recovered. In 2015, with the cauliflower coral in particular, but also other species, it stayed warm too long. So the bleaching took place and the corals died. And now they're in place with seaweeds starting to grow on them. And if those herbivores aren't there, they're not going to come back. Once the seaweeds take over, you're never going to see coral again unless something removes the seaweeds. And that's why these herbivores that we study, these grazers, are so incredibly important. Whether this experiment goes properly or not, we know the grazers are important. We're just trying to figure out, are there ways we can enhance their abundance locally? Yes?
How long is too long?
For the corals? So that's a function, that depends on how warm it gets. So scientists have made some graphs that show, you know, if it gets, there's kind of a trade-off. If it gets way above normal, they will die sooner than if it gets just barely above normal. Days to weeks, I would think, would be the range.
Go back to before the first slide.
Before the
Talk
what?
about global warming and the temperature.
Yeah,
But the oceans are not necessarily in sync with that. There are places, there are mountains of hot water and places of cold water. Right now, I think Hawaii is sitting in a relatively hot sub-ocean. Those places, many of those are in sync with climate.
I'm not sure I understand the question.
The temperature of the ocean is not constant.
The temperature of the, yes,
It
it's
goes
varying.
depth and also goes horizontally.
That's true, yes.
And there are places where warm water accumulates long-term atmospheric conditions too.
Yes.
Now, those necessarily some in sync or not in sync with global warming.
Well, when I think of the word in sync, I think of,
Always going up with the, going up together.
so if you average the whole ocean, it's going up. But at any given time, there are warm places and cool places, and those are dynamic.
Point right here, how is the ocean water close to the island, not the near shore? How's that temperature changing with climate change?
In recent decades, Hawaii has been lucky to be in a part of the ocean that's not warming as rapidly as elsewhere in the ocean. But when we have a strong El Nino year, or when we got what was called the blob, this huge parcel of warm water that developed in the North Pacific Ocean, and that impinges on us, that's when we get our coral bleaching events. So this is why the oceanographers are now projecting forward and saying, okay, given that there is this variation, warm water, cold water sometimes, warm water sometimes, as the whole system warms, how frequently are we going to be getting bleaching events? Because it just takes 1 degree centigrade over the summer maximum temperature to get bleaching to take place. And that's where they're coming up with right now, global average every 6 years. By 2040, every year. Doesn't matter how much variation there is. All the variation will be above that threshold within a couple decades. Yes.
The first picture you had was all white coral. What caused the bleaching?
Well, that's those little single-celled plants leaving the coral. So the coral polyp itself is like a little sea anemone, and it has no color. It's transparent, and its skeleton underneath is white. It's calcium carbonate. It's limestone. So without those single-celled plants in there, all you see is a white thing. You see the coral skeleton looking through the transparent polyps that are the actual living animals. So what we call bleaching is actually just the coral without all those little single-celled plants inside of it. They've been expelled.
I think that coral, I'm not looking.
You're not supposed to.
You have a question?
Helen.
Where are eggs? Or is are there any active? I know like the resurrection ponds and like that. Are they still found in certain places? Like we find units, or are they any that are being restored?
What I have read about them in Hawaii is that there are remnants along the windward coast, including Hanauma Bay. They were typically used in areas where there was no living reef, where an ahupua'a was not blessed with a nice active reef. So they built these little sort of basically little tiny artificial reefs out on the sand flats to bring the fish. And there's evidence of remnants of these here and there. They're also very common throughout Polynesia. I know that they're still actively used in Samoa and elsewhere. I don't know. What I read was a very limited account, and that's all I've been able to find so far. I've been trying to find a Hawaiian scholar who knows more about them. But I feel honored to be using an old tool for a new problem. I'm sorry, I can't hear you.
Do you have any photos of those modules?
Of the closed modules? Not with me. They're not particularly interesting, which is why I didn't bring them. But I go back to this. They sort of look like that, except there's no holes. So, and that's, again, that was unusual. We thought, we expected, oh, well, if they don't have holes, they'll be covered in seaweeds. And if they do have holes, they won't. But it depends upon where you are. So in Waikiki, the ones without holes look just as fuzzy as these ones. And the ones without holes in Hanauma Bay look just as clean as these ones with holes.
Some of the fish ponds have something akin to this, piles of rock, lots of cookers in it, so little fish would have places to hide.
Yeah, those are the emu.
And was there a name?
The name?
Yeah.
Emu, I-M-U. Sometimes I've seen it spelled umu, U-M-U, but mostly emu,
Yeah.
I-M-U. Yeah.
As far as the uhu, are there any, like, that if things break, so bring them back, the fisheries?
Well, the most effective way to bring back the uhu is to stop fishing them. The problem is they're extremely popular as food fish. And unfortunately, we do not enforce our fishing regulations in Hawaii. And what very commonly happens is because uhu sleep in holes at night, they're inactive in holes right under ledges. Guys will go out scuba spear fishing at night and just wipe out a whole reef. They're sitting ducks. They're just sitting there and just bang, 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 bang. And they even have a name for it. They call it bombing a reef. We bomb that reef, and I could show you photos that would sicken you. 3 guys with 3 dozen uhu in front of them that they speared in one night. So unless we start regulating the consumption of uhu, we're kind of doomed. And that's one of the reasons we wanted to do this experiment in 2 different settings, because I doubt if we're going to see any uhu in Waikiki, no matter how many holes are out there, just because there's such intense fishing. But we also need to see what happens in Hanauma Bay, where fishing is regulated. So the scientific community right now is working with the state for the state to see the importance of herbivores for the future of our reefs. And I don't know exactly what the state's going to choose doing. The alternatives are gear restrictions, say no scuba spearfishing at night, or spatial management where there's no fishing in these areas at all, or just no fishing uhu, no matter what. But regardless of the regulations, there has to be enforcement. And right now, the state of Hawaii has no money for enforcement. So right now, it's really a free-for-all, unfortunately. Sorry, you got me on my soapbox.
Mm-hmm.
Yeah, didn't the state just block the aquarium trade for good? Is there a permit with that? I live in Indonesia, but I was reading about it.
Yeah.
That's great.
Well, yeah, the aquarium trade, it's really interesting. Most of the aquarium trade in Hawaii is done on the Big Island, on the Kohala Kona coast. And it targets primarily 2 species, yellow tang and kole, 2 surgeonfish, 98% of the catch. It's extremely well regulated. The Kohala Kona coast, a third of that coast is covered in 9 fisheries replenishment areas where there's no aquarium fishing allowed. And since those went into place, brought forth by community-based management, the fishery has thrived. And so the fishery really isn't causing problems anymore. However, when people are out snorkeling and they see people put up a wall net, catching all these little fish, it upsets them. And so there's been a very strong movement against the aquarium fishery. But the irony is that there's no enforcement of regulations in Hawaii except for the aquarium fishery, which happens to be perfectly well regulated by management. So it's ironic to me.
Yeah, I was thinking the next step would roll over into the food.
Yeah, I would love to see. I would love to see enough kupuna and kahuna step forward and issue a kapu on uhu to the populations recover. I would love to see that. We need those fish if our reefs are going to survive. And right now they're almost gone. I lived here in the 1970s, and just between then and now, it's just astonished me how few parrotfish we see. And in the back row, there's a question, yeah.
I know Maui at least, they have these areas where they have specific herbivorous fish that you are not supposed to take.
Mhm.
Literally have signs saying, don't take
Yep.
fish, don't take these here. But if you want to take tape, if you want to take alloy, go ahead and steer them in there. And they had that, I think, at Old Airport Beach.
Yeah, the one I know of is Kahikili on Maui. Yeah, and that's been in place a while now, and what we've seen is that the uhu are coming back, and the surgeon fish are coming back, and the algae is declining in abundance, and a special type of algae that's called crustose coralline algae is starting to grow more, and corals love that stuff for settling. The corals haven't come back yet, but the system is heading back toward health. And so it shows that if an area is closed, just don't fish the herbivores or the grazers, then it can recover. And that's why one of the things that DAR is, the Division of Aquatic Resources, is considering now is setting up a network of herbivore replenishment areas around the entire state. My fear is that we'll still lose the reefs outside those areas. So if we cover 10% of the state, we're going to have 10% of our reefs left in the end. I really think we need uhu everywhere. Happy uhu, big fat ones. Mm-hmm. The scientists call them boffs. Big old fat fertile female fish, pushing out lots of eggs and eating lots of algae. Yes.
I just remembered what the name of urchin is. It's called ha'uke.
Ha'uke. I wonder which species that is.
Pencil or not.
As I said, we just shake everything up.
The
Oh.
purple one. Little purple one on the show.
No, it's not purple.
I'll use that.
Yeah, I just don't know them all. This is why when we were first talking about the title, we were going to say uhu and whatever word we were going to put for urchins, and we gave up.
I said it's safer to just go with parrotfishes and urchins, sea urchins, because there are many different Hawaiian names for different species of sea urchins. So let's just stay safe.
Stay safe.